Okay, I'll start with a short introduction and then I'll hand over to Professor Wall and Professor Preddy that will talk about this new study that has been published and it's available on the Rare Connect TMAU community. So, welcome to the TMAU Community Webinar. My name is Marta Campabadal and I work with the European Organization for Rare Diseases as an online community manager. Today, we are very pleased to have Professor Preddy and Professor Wall from the Monell Chemical Sciences Center, a research institute with a program to evaluate people with other complaints for TMAU. During this webinar, we'll have a chance to hear from these researchers and ask them questions about their work and how it could benefit TMAU patients. Today's webinar is based on the results of a study about genetic analysis of impaired trimethylamine metabolism using whole exome sequencing. The full text, as I said, is available on the Rare Connect TMAU community. Before I hand over to Professor Preddy and Professor Wall, I just want to show you some numbers about the TMAU community on Rare Connect. Here it is. This is the link where you can join the community. This community is one of the most active communities on Rare Connect, with over 2,050 users, 10,000 visitors per month, and 1,600 forum conversations with thousands of comments in different languages from members from all over the world. Today's webinar will be the 5th TMAU webinar and it will be uploaded on the Eurodis YouTube channel along with the other 4 that are already there. If you have questions, please write them at the chat box that you'll find on the left side of your screen, and we'll try to answer them at the end of the presentations. So thank you very much. Now I hand over to Professor Wall that will start the presentation about this study.
So hello everybody, I'm going to discuss the TMAU genetics in this webinar. So first I will introduce the background in human genetics, just in case you are not very familiar with this, and also I'm going to talk about genome variants and methods to detect them. And then I will go through the rare Mendelian disorders and finally the TMAU genetics analysis. So this is a schematic graph of the cell of a human. So you can see there are multiple organelles, and a cell is smallest structural unit of organism, and it have a lot of functions. And then so cells store all information to replicate itself, and there are 3 billion base pairs, the basic unit of the human genome. And almost every cell in human body contains same set of genes, but not all genes are used or expressed by those cells. There are also other machineries in the cell, which I won't go through very in detail. So there are multiple key terminologies in the genetic study. So the first is the genome. Genome is an organism's complete set of DNA, the genetic material. And so for humans and mouse, those 2 genomes have almost 3 billion units, the basis. And human genome has 24 distinct chromosomes from the autosomes number 1 to 22, and then sex chromosomes X and Y. Each chromosome contains many genes. So what is a gene? Gene is a basic physical and functional unit of heredity of genetics. It is composed of specific sequences of DNA bases that encode instructions on how to make proteins. And there's also the genotype. Genotype is the genetic makeup of an organism, and so there's the opposite of this is phenotype. Phenotype is the physical expressed traits of an organism, such as your height, your weight, or your disease status, and etc. So genotype and phenotype are a pair of definitions that are the central, the central stuff in the genetic study. So we are trying to find links between genotype and phenotype. And also there's the definition of nucleic acid. So it is the biological molecules, for example, RNA and DNA, that allows organisms to reproduce. And finally, the proteins. Proteins make up the cellular structure, and it is large, complex molecules made up of small subunits called amino acids. And then we come to the three basic critical molecules of the life. So they are DNAs, RNA, and proteins. DNAs are the basic genetic material, so it holds information on how cell works. RNAs act to transfer short pieces of information to different parts of the cell, and also provides templates to synthesize into protein. Proteins form enzymes that send signals to other cells and regulate gene activity. And proteins also form body's major components like hair, skin, your bone, or other organs. In 1953, those two scientists found the DNA's double helix structure, and they won the Nobel Prize for physiology and medicine. And also, DNA, excuse me, DNA is a linear structure of AGCT, those four letters. And you can see DNA is inherited from each of the parents. So there are two parental strands like this. And also there are daughter strands that are replicated from this parental strand. The structure and the four genomic letters code for all living organisms in the earth. And also the adenine, guanine, thymine, and cytosine, which pair AT and CG, those pairs on complementary strands. So this is the central definition of DNA. And there's the central dogma of molecular biology, which is a information flow from DNA to RNA and then to protein. DNA can also replicate itself. DNA to RNA is the information coded passed to molecules of RNA, and then RNA can be translated into protein to function. This is the genetic codes for protein. For example, in the very middle of this figure is the first letter of the genetic code, and then the second letter, and then the third letter. So each three letter of DNA is called a codon, and then this codon corresponds to the RNA letters, and then to protein letters. So all the 20 protein letters are encoded this way. And then after RNA form protein, there's also the post-translational modification and protein folding, which is complex and can form various functional components. What makes us human? So the genetic material are coded into the chromosomes, and it looks like this. We can use karyotype to study those chromosomes. So it can be divided into autosomes from chromosome number 1, 2, 3, and all the way through 22. And there are also sex chromosomes, the chromosome X and chromosome Y. So chromosome Y is only found in a normal male person, but there are some other abnormalities of chromosomes that contain different sets of chromosomes. And in 2003, the Human Genome Project finished, and it is the milestone of this genome era. So we can analyze human genes and genomes in a very systematic and efficient way. So the strategy is we have a reference which is sequenced 10 more years ago, and then we compare the individual's genome to the reference genome to find variations or variants. Some of them are called single nucleotide polymorphism, or SNP, or single nucleotide variant, SNV. And some of them are small insertions and deletions to the sequence of DNA. They are called indel. Most of them are very short, for example, less than 10 BP. BP is the unit of the DNA. And there are also copy number variant or copy number variation. So they are different copies of the stretch of DNA. For example, we have 2 strands of DNA, so the copy number for a normal chromosome should be 2. But some of the regions of the genome are duplicated. For example, we have, 3 copies of the same region. So this is a copy number variation. And some of the parts are deleted. For example, only 1 copy of the region or 0 copy of the region is there. So that's a deletion. It is also a type of copy number variation. There are also other complex structural variations in the human genome. For example, translocation, which you have a switch of 2 regions of the genome, or inversion, which is the total inverted region. So, but the meaning of normal or wild type variation or wild type genome is very relative. It is just regard to the diploid reference genome. The most frequent variations are biallelic SNPs, just like this. So if the normal or reference DNA sequence is like this, the ATC-TAG, and in a variant or mutated DNA, it is ATC-GAG. So the middle T is mutated into G. So this is a SNP, or a single nucleotide variant. This can affect the downstream RNA and protein sequences. And why they are called biallelic? So because in the population, most frequently there are only 2 versions of this allele, the T and the G, so they are called allelic. And there's a major allele which is more frequent in this population, and there's the minor allele. For example, this mutated G. The minor allele is less frequent in the population. And the single variants, they can be good, they can be bad, and they can be silent. So single variants can serve the organism in 3 ways. The good ones can cause a trait that enhance the organism's function. For example, the mutation in the sickle cell gene provides resistance to malaria. The bad ones can cause a trait that is harmful, sometimes fatal to the organism. For example, Huntington's disease, a symptom of a gene mutation, is a degenerative disease of the nervous system. Sometimes, or actually most of the time, the single variants are silent. For example, they do no harm, they do no good, but and there's no difference in the function of the organism. The vast majority of human genome variation fall into this group, but whether a SNP or SNV is silent also depends on the population. And there are recently there are a lot of technologies to detect variation in the human genome. The first kind is called genotyping. It is microarray based and use hybridization, and it is highly automated. A lot of companies are doing it. For example, Illumina, Affymetrix, and so on. The characteristic of this type of technology is the SNVs or variants are predefined in those technologies, and it range from a few 100,000 to several million markers. Another advantage of this technology is you can also detect CNVs, the copy number variations, using this technology. Recently, actually 10 years ago, there's the GWAS array, which is for genome-wide association studies. So this kind of array, genotyping array, contain common SNPs that are with 5% or more minor allele frequency in the general population. There's also the gene-centric arrays focusing on specific regions as opposed to the genome-wide, which is across all the genome. But this gene-centric array is centered on the candidate genes and can also contain uncommon and rare SNPs, which have lower minor allele frequency, the MAF. There are also the traditional Sanger sequencing method to detect variation. The procedure generally is primer design and then performing PCR and then put it into the microfluidic capillary sequencing machine and then got the chromosome chromatograms. This type of method is semi-automated and has the length limit of around 1000 BP. So you cannot do it in the whole human genome because the human genome is like 3 billion BP. If you do this, it will be take an enormous time to do it. But this method can detect novel variants because it is a method to sequence the whole length of those 1000 BP instead of just looking at the predefined variants for the genotyping method. And about 10 years ago, there's a new technology called next generation sequencing. So actually, the development began in early 2000 in Cambridge, and then Illumina, the company in California, bought this company, the Solexa company, and then developed the high-seq machines, which is a highly automated and high-throughput machine to sequence those variants. And there are also other companies like LifeTech, Complete Genomics, Roche 454, and other like PacBio and Oxford Nanopore, and those companies are also developing new methods to sequence the human genome. So now the next generation sequencing have two major applications. One of them is whole genome sequencing, where you sequence all the human genome using this method. The other one is whole exome sequencing, where you have the coding regions of the genome, which comprise the like 1% of the whole genome. So those two applications are widely used and are in the clinical setting as well. Whole exome sequencing and whole genome sequencing don't assume any prior information, so you can find new genes, new variants using these two methods. And whole genome sequencing is kind of straightforward, but for whole exome sequencing, you need an extra step to capture the exome or the coding regions of the genome and then sequence those captured sequences. Next generation sequencing is high throughput, high accuracy, low cost, and generates a large amount of data. So typically the data is in like terabytes. So it's a large number of sequences and data. So it is computationally intensive. You need to use Linux or a Unix cluster or cloud computing to analyze and process those data. So now I'm going to discuss a little bit of the human genetics, especially the Mendelian disease. Mendelian inheritance is is the basic of medical genetics. So there are 5 or 6 basic inheritance models. For example, the autosomal recessive, like in this part. So you can see, for example, those two the parents in this category are consanguineous, then it's highly likely that some of the offsprings are affected with autosomal recessive disease. Or there could be the compound heterozygous mutations in the offsprings of this pedigree. So it is also a form of autosomal recessive disease. And then there's also the category of autosomal dominant. For example, in this case, so this family, the first generation, the father had have the disease, and then it inherit, and then the disease transferred, transmitted to the offsprings, and then to the second and the third generation. And there's also the de novo model of the autosomal dominant inheritance. For example, in this case, so neither parent has the disease and no mutation in those two parents, but the child has a de novo mutation which is new to either either parent, and then this is the de novo model. And also there's X-linked recessive and X-linked dominant model, which I won't go into detail. And there's also the Y-linked model and the mitochondrial inheritance. Keep in mind that there are exceptions to these inheritance models. For example, there are variable expressivity and incomplete penetrance to the pedigrees. Mendelian diseases are caused by single mutations, mostly in the coding region of exons, thus alter the protein structure and function. And that's why we can use the whole exome sequencing, which interrogates the coding region of the genome to, the coding region to detect the variants. There are the traditional method of linkage analysis to find genetic markers of the rare diseases. The method is trace the mutation across multiple generations. There are shortages of this method. For example, it is low resolution due to marker size. So the traditional markers are over several hundred or over 1,000 BPs, and it also requires large multi-generational pedigrees such as this one. So for example, this one is a big family of inherited disease, and the disease is partially resolved actually after 60 years, 60 years after the original report on this part, because okay, yeah, so it should be like this, yes. So the original part is within the dashed lines, and then after 60 years, there are offsprings of this original family, and then the researchers can find the responsible gene for this disease. But this is for the traditional linkage analysis. And for now, we have the NGS-based whole exome sequencing method, which scans all the coding regions of the human genome in a very detailed base pair resolution, which is a very high resolution. And we don't have to use large families. Small families are also fine. For example, a trio, the father, mother, and the affected kid. And even multiple sporadic cases are also fine, such as TMAU. We don't have family information for those patients, but we can compare the genetic information of the sporadic cases for TMAU to find some commonalities between them. The general procedure of NGS-based Mendelian disease sequencing project is you get the raw FASTQ data, the short reads of the NGS platforms, and then you align, next slide, and then you align or map them back to the reference genome using softwares. And then this is to the right is an illustration of the sequence of the region that that we have data for the whole exome sequencing method. You can see it is sequenced multiple times for each individual. So every chunk of the data, every chunk of the map is individual. So you can see in the middle, the first individual have a mutation. So if it is different from the reference genome, it is indicated here. So the first individual have an A here. So I don't know the reference, but it is mutated here. And then in the following 1, 2, 3, 4 individuals, they are all mutated in almost half of the sequences. So every line, every row in the figure is a short read of almost 100 BP. And then you can see it is deeply sequenced in this position for each individual. That's why the method is also called a deep sequencing. So after you have the alignment like the figure in the right, you can call variants using softwares like a GATK. So this is the general procedure of the NGS pipeline. So first, the samples are put into the NGS machine, and then you can have raw sequencing data in the FASTQ file format, and then use BWA to align those reads, those small pieces of DNA to the reference genome to have alignment results called BAM files. And then you can use GATK to detect variants in the format of VCI files, and then and then you can use frequency from public domain and internal databases to filter those variants to get rare variants, and then annotate those variants to have the ones that are in the coding regions that are deleterious or harmful to the proteins. And then you can also use the non-synonymous and the deleterious range to finally got the variants here. Finally, we can use the biological information or disease databases such as HGMD, the Human Gene Mutation Database, and OMIM, the Online Mendelian Inheritance in Man, to filter variants that are relevant to the disease you are looking for. Now I'm going to introduce a little bit of the rare disease sequencing project, which is called RD1000. So it is a large rare disease sequencing effort in progress, collaboration between BGI, CHOP, and multiple sample sources from US as well as the world, around the world. And we share phenotypic information, the categories, diagnosis, medical records, disease-specific knowledge, and we also share genotypic data. So most of the DNA are sequenced by BGI. CHOP also sequenced some of the samples. And we share authorship and opportunity to grant applications and intellectual properties. And actually, TMAU, the project we are talking about now, is part of RD1000. So it is the so RD1000 is the mother project of the TMAU genetics study. So now I'm going to okay. So this is different from what I expect. All right, so I'm going to briefly introduce the TMAU. So it is trimethylamine has unpleasant odor of fish, and so if you eat choline and then the gut bacteria turn it into TMA, and then TMA is turned to TMAO, so which is odorless. But you need FMO3, the human liver enzyme, flavin containing monooxygenases 3. So you need this enzyme FMO3 to turn TMA to TMAO. But specific variants in the FMO3 gene are related to impaired TMA metabolism, and genetic variants of FMO3 can inactivate or lower the oxygenation efficacy. Yeah, so there are more in this slide actually. And FMO3 variant, but the FMO3 variants don't explain all the TMAU cases. So there must be other genes contribute to this process, especially those within the same or similar biological pathway of the FMO3 enzyme. Oh, this is all messed up. All right. So thus we conduct the current study to investigate the genetics of TMAO. First, we test the order. We did the order evaluation by a trained sensory panel, and then we did the analysis of their urine concentration of TMA relative to TMAO. So this is there's a reference of 0.9. If the ratio is below 0.9, then it is diagnosed as TMAU. And last, we did whole exome sequencing as well as a subsequent variant analysis of all the 10 samples we sequenced. This was published in BMC Medical Genetics like 1 month ago. Okay, so this table lists all the 10 patients we collected and analyzed. So we informed the other subjects with body odor complaints who contacted the study investigators about participating in our ongoing sensory and genetic studies. Approximately 130 subjects replied, and we choose 10 of them for whole exome sequencing. This table is ordered in the TMAO to TMA ratio. You can see the most severe one is of 0.13, which is far from the 0.9, which is the cutoff for the TMAU disease. And there are also two ethnicities of this study. So at first, we look at the genetic variants in known genes, namely the FMO3 gene. So there should be a figure.
Yeah, I think you added of FX on this slide.
Yeah.
Is that right? Yeah, I think that's why it's not working because the effects on PowerPoint mess up with the webinar room.
Okay, so can you?
Just skip and yeah, move to the next one, to the next slide. Is that okay?
Actually, can I share my screen so that everybody can see
Yeah,
my slides?
you can try.
Yeah.
Okay.
So can you see my screen or?
No, but I'll stop. Can you try now?
So how can I share my?
Do you see do you see a share my screen button somewhere?
Oh yeah.
Okay.
Oh, let me see this one.
It's loading. Give it a second. Okay, I see nice mountains.
You can
Okay.
see it, right?
Yeah, perfect.
Yeah. Yeah. So at first we have this. Those are the FASTQ files, the short reads of 100 BP, and then we can map them or align them back to the reference genome. So these are the alignment files, and then this is the variant files, which we called variants. You can see those are all computer analyzed data, so we need a bioinformatics support. And then I have this table of known rare and common pathogenic variants for TMAU observed in this study. So those variants are in the known genes linked to TMAU. We can see there are 4 FMO3 variants. The first one is very rare because the MAF is very low. It's in the 10 to the power of -5 level, which is very low. And then and this mutation is actually causing amino acid change, which means the protein is changed due to this mutation. For example, here glycine is changed to a arginine in the 148 position of the amino acid chain. So this is found in subject 99, one of the samples in our study. We also found 3 FMO3 variants that are not very rare because their MAF is not that low. So you can see those 3 FMO3 variants are somewhat common. So they are common in population, and there are established reports that are associated with them with impairments in metabolism of the TMAU. So those 3 variants are also important in the pathogenesis of TMAU. And last, we also found a PYROXD2 variant. This is a intronic one and also have high frequency in the population. And after that, we tried to find variants in genes that are unknown to cause TMAU. So for the previous one, we just filter the mutations, but for this one, we need to have a little complex procedure. This is a workflow. So we called entire. So we have the larger sample size. Remember, we have the whole rare disease sequencing project with almost 700 samples, and the TMAU is just a part of that. So this is the entire large sample we have. We can pile up all those samples sequenced and then find the SNPs and the indels. So those are the numbers. And then we find homozygous for a minor allele, because minor allele, generally speaking, minor allele is harmful to human. And if it is in a heterozygous status, it is mostly fine. But if it is in homozygous status, that means you have 2 copies of the minor allele, which is more severe than the heterozygous state. So we try to find all those homozygous minor alleles. And then we got the result of 94,000 SNPs and 5,000 indels. And then we used a software to predict whether those variants are pathogenic or whether they are harmful to the downstream proteins. And then we got 1,800 SNPs and 163 indels. And from there, we used 2 strategies. The first is we try to see if there are any variants in the oxidoreductase pathway. The other one is whether some of the variants are shared by 2 or more patients. So if 2 patients share 1 variant, so this variant is more likely to be the ones that are linked to the TMAU as opposed to the ones that only 1 patient has. So for the in the oxidoreductase pathway, we finally have 57 SNPs and 4 indels. And after the frequency filtering, for example, we use the MAF of 0.05 in the general population to filter these results. We finally have 4 SNPs and 1 indels. Follow the same procedure, we have 9 SNPs and 1 indel for the variants that are shared by 2 or more patients. So here is the result. So in the upper part, those are the variants that are found in oxidoreductase pathways that are with more biological support of the genes. So you can see at least one of the 10 subjects was homozygous for the alternative alleles and predicted to be harmful. And with MAF less than 0.05, MAF is minor allele frequency. We observed that 3 subjects, number 52, number 122, and number 114, are among the lowest TMAO-TMA ratio. So those 3 patients are in the extreme of the TMAU phenotype. And we also found 9 SNPs and 1 indel using the sharing strategy. So all of them are shared by 2 or more patients. Actually, shared by 2 patients. For example, this variant is shared by number 62 and 99. And there are also genes annotated with these variants. And we also used a gene-gene interaction strategy to find variants in the interacted genes. And I can show you the interaction network in the next slide. So like this. So for example, FMO3 is in the middle, and you can see there are multiple genes interacting with that. And the evidence is based on literature searching and co-expression, and also other binding and other experiments. And for the PYROXD2, we also have a network, and for DMGDH, so which is previously reported to link to TMAU as well. So we found 3 variants in the interacting genes of those 3 genes. And we confirmed those variants by Sanger and Techman sequencing, showing a concordance rate of 100%. So that means the whole exome sequencing strategy we used is very accurate. So we confirmed or validated all of the findings we have. And for future direction, more study is needed, and we are seeking funding opportunities to test DNA of another 100 TMAU patients in order to identify new genes involved in this disorder. So for acknowledgement, I want to thank colleagues in my lab, the Center for Applied Genomics at the Children's Hospital of Philadelphia, especially Director Hakan Narson. And also I want to thank the collaborators at Monell Chemical Synthesis Center, George, Dr. Daniel Reed, and Corinne, and Daniel, and others. And also thank BGI for their sequencing, their continuous collaboration in rare disease research. Thank you. Hi, George. I have a question. So, do you think it is possible to have some normal FMO3 to like to how to say neutral this disease? To take
You mean introduce it somehow?
FMO3 and then.
Yeah, you would have to you would have to protect it though. I mean, if you want to, how would you introduce it into the? You could introduce it into the digestive tract, but most of the digestive enzymes would chew it up. So it would have to be coated in some way to make its way down to the large intestines where all the trimethylamine is being produced. So unless you did that, you would probably fail in getting any type of effect.
Thank you.